All right, welcome everyone. Today we're going to be talking about regulatory circuitry and networks. So again, it's the last part of the first module. So first we're going to talk about regulatory motifs and gene regulation. Then we're going to talk about how we discover these motifs, how do we use comparative genomics to find these motifs, and how do we use evolutionary signatures to find individual motif instances. And then we're going to pivot to, now that we have these networks, how do we analyze networks? So the basic building block of gene regulation is basically the regulatory motif. This is allowing us to understand the molecular instantiation that is underlying these edges. So every time we think of regulatory edges of regulator A controls gene B, what you're going to have is an edge between that regulator and a physical location on the genome that this regulator binds. And that physical location is usually showing a common sequence. And that common sequence is what allows the regulator to bind. Everybody with me here? So these are the building blocks of these regulatory edges that we're going to be studying in the networks part of this lecture. And these edges can be found within larger regions, which allow them to be bound, because these larger regions compete against the nucleosomes to exclude them. And for that competition to work, usually multiple motifs help each other. So you can think of this as a biophysical model of binding, which is purely based on physics. No fancy, oh, I'm talking to this guy and we're collaborating, etc. No, it's just every regulator has some probability of binding DNA and just being detached, binding and being detached. And if there are multiple landing sites for multiple regulators at the same time, then they're going to be helping each other fight against the nucleosomes. Is everybody with me here? So they're going to be keeping the region open so that purely based on biophysics, they can kind of help each other. Now, in addition to that, you can add protein-protein interactions of these factors, which are basically binding, one detaches, the other one is still bound to it, and so on and so forth. Is everybody with me here? So that's the reason why regulation happens in these regions rather than with isolated motifs. And a lot of work by our lab and others has basically shown that motifs that are sitting in the middle of nowhere are much less bound than motifs that are sitting within these regions. Everybody with me here? Good? All right. So now how are regulators binding? So, if you're going to recognize TAA, TTA, what are you going to do? You're not going to open up the DNA and then sort of by complementarity read it. What you're going to do is that you're going to feel it from the side. The DNA itself doesn't open. The double helical ladder is still closed. But if you have an A base, yes, it matches a T based on the contacts on this side, but it also can be recognized as an A from the side. Everybody with me here? So basically, AT bases have contacts with each other, GC bases have contacts with each other, but they also have additional atoms that are basically recognized from the side. And that tells you a little bit about the structures of the sequences themselves. If the protein is binding a single side of the protein, of the DNA, then it will be one contiguous motif. Everybody with me here? If it's binding two different sides, then it will be two cores separated by sometimes fixed, sometimes variable spacing. Is everybody with me here? And then if the protein itself is a homodimer, in other words, if it dimerizes, if it sort of binds together with itself, then it will be a palindrome, which basically is a word that is read in both ways, front and back. Pali means again, drome means road, so you take the same road again. But it will be a reverse palindrome, namely GAA will be read as CTT on the other side. And the reason is because of the complementarity of the strands. Everybody with me here? Yes? Awesome. So basically, that's the building blocks of gene regulation. And these motifs are basically coming in many shapes. You can see here that this one is a reverse palindrome, TAA, TTA on the opposite strand. Same here, CACGTG on the opposite strand. And this one is not, so GATA, okay? And then the structure of these regions is, the structure of the motif is inferring the structure of the binding of these regions. And when you have regulatory variation that disrupts these motifs, these functional instances, they will be manifested as disease-associated variation some of the time. Everybody with me here? So basically, there's going to be mutations all over the genome. Tiny subset of those mutations are actually impacting functional elements because the vast majority of the human genome is not actually functional. But the fraction that disrupts proteins will do so by disrupting an amino acid, and the fraction that disrupts regulation usually will do so through one of these regulatory motifs. Everybody with me here? And in the genetics portion of the class, I'll tell you about this extraordinary example that we uncovered in collaboration with Melina Clausentzer that basically shows that this conserved regulatory motif is basically disrupted by a genetic variant associated with obesity. And that basically leads to an extra 10 pounds in a homozygous adult just by one letter change. By disrupting this motif, no longer binding this repressor, derepression of these genes, and then alteration of the core process of thermogenesis, of heat generation, that allows you to, instead of storing calories, burning calories. So homozygous individuals for a C disruption of this AT-rich motif can no longer burn calories. Everybody with me here? And then this was published in the New England Journal of Medicine back in 2015. So this is what we're tasked with today. How do we discover these motifs? How do we infer these edges? And how do we use the resulting networks to understand biology? Everybody with me here? Awesome. So when we talk about motifs, I want you to distinguish the concept of a motif versus an instance of that motif. A motif is a generative model. This sits in the model part of the world. An instance is one instantiation pulled from that generative model. Everybody with me here? So a generative model basically says, here, I will generate characters in a position-independent way, and I will sample from an underlying distribution of A or G, equally likely. Both are good. C or T, both are fine. C with, maybe 3/4 probability and A with 1/4 probability. Is everybody with me here? So, and then this better be a TCA, otherwise I'm not going to like it. Sounds good? So you can discover these motifs and represent them as position weight matrices, or PWMs. And these PWMs allow you to basically say at every position, what is the frequency with which I observe different nucleotides in all of the, for example, bound instances of this regulator. So for example, this ABF1 regulator has all of these bound instances that we found experimentally, and then you can kind of line them up. And then what you see is that there's a very conserved TCA here with either A or G in the front. There's a very conserved ACG here with sometimes an A, sometimes a G following it. And then the middle positions are free to change. What's the shape of the binding? Very easy. It's something that sort of binds the two parts of the helix with a small spacing in between. Everybody with me here? Awesome. Good. So again, distinguish the concept of a motif versus an instance, and the assumptions that such a representation inherently makes is that there's independence of the different positions. In other words, every time I have an A here, it tells me nothing about whether I should also have an A here. Okay, so that assumes independence of the different positions. It assumes also fixed spacing. And the way to overcome that would be to say, well, if there is dependence, for example, if the A only occurs with a C, one way to represent that would be to take this sequence and to split it into two motifs. To basically, it's either fixed A, fixed C, and all of the rest, or fixed G, fixed C, fixed D, and all of the rest. Does that make sense? So I can basically take a position weight matrix and split it into two position weight matrices that capture those dependencies. Everybody with me here? I can also do the same thing for variable spacing. I might have 5 or 6 or 4 nucleotide spacing if there's flexibility in how I'm binding the DNA. And then same thing, I can just split it into one version with a 6 gap, one version with a 5 gap, one version with a 4 gap. Can I get a 5, 4, 3, 2, 1? How well you're following so far? Awesome, bunch of 5s. This is great. All right. So again, motifs are acting very often at the DNA level, but some of the time they're acting at the RNA level to decide if a particular transcript will be spliced or not. They can also act at the protein level to basically see if I'm going to glycosylate or, bind something with a kinase, or use this to target my protein somewhere, and so on and so forth. So basically, you can think of the same exact methodology for these motifs at the level of DNA, RNA, protein, and also just physiology while we're at it. And, you know, I don't know, joke patterns of your professor. Like, you know, there's like some kind of motif that you can recognize, and you can sort of find recurrent patterns, of course, in your schedule or in, you know, I don't know, the cleanness of the status center and so on and so forth. Everybody with me here? So any kind of recurrent pattern can be represented in such a position-specific motif. And there's many ways to discover motifs. We're going to talk about expectation maximization and Gibbs sampling. You can also simply enumerate motifs. You can basically say, well, I'm going to search every TCA, you know, with whatever letter afterwards, and you can kind of simply enumerate. You can use evolutionary conservation, and you can also use information about the domains of the corresponding proteins. You could be like, well, this is a helix loop helix domain, or this is a, you know, zinc finger domain. And then there's specific patterns of sequences that can be associated with the binding of the corresponding protein structures. Everybody with me here? Awesome. Good. So there's experimental ways of discovering motifs. One way is you could basically have some kind of selection that allows you to use a protein target of interest. You have different sequences. They can be DNA or RNA, and then you can kind of pull them out. And then the ones that you that you pull out, you could enrich them further, and then do another selection, another selection, another selection, progressively weeding out all of the sequence fragments that are not bound by any of these proteins. Okay, so this is an experimental way to enrich for actual DNA fragments that are pulled down by a particular regulator. Everybody with me here? Good. Another way is you could basically build a microarray chip where you synthesize these fragments of DNA, and then you wash through whatever protein you care about, and then you see whether the signal from that protein, for example, fluorescence or you name it, is happening at specific locations. And then that'll tell you that the locations that had that pattern are in fact preferentially binding whatever protein you washed through your DNA microarray. Everybody with me here? Yeah, good. And then you can also use, you know, protein binding microarrays, etc. But in general, the three big concepts that I want you to think about is number one, the motif itself, and that's the generative model that creates instances of that motif. Then the individual instance of that motif. And then the regulator that can be physically bound to a given instance of that motif, and that has an affinity for the generative model of the motif. Okay? So these are the three major classes of elements. And then the question is, how do you discover the regulators themselves? How do you discover the motifs? How do you discover the individual binding sites? And if you have the regulator, how do you find its motifs? If you have a motif, how do you find the regulator? If you have a regulator, how do you find all of its targets? If you have the targets, how do you find the regulator that's bound to those targets? And if you have a motif, how do you find all of the instances? And if you have a bunch of instances, how do you discover the motif? Okay, so these are the main challenges, if you wish, in regulatory genomics. So to discover the regulators themselves, you can basically look at homology to other regulators. And then you can say, okay, well, that protein is probably a regulator as well. Let me go experimentally test it. To discover the motif, you can basically use comparative genomics, find conserved islands, as I showed you in lecture 2. And within those islands, look for common patterns. And then you will be able to de novo discover these motifs. When you have a motif, you can find its targets by simply matching that sequence. But the vast majority of the matches will not be functional. As I mentioned earlier, you need a certain context of the chromatin for the motif to be successfully recognized. Are you guys with me here? So that basically means that I will need some additional information to go from the motif to the target. For example, I could be looking for preferential evolutionary conservation of these targets, because those targets that fit within enhancer regions, promoter regions, and other functional regions will be preferentially preserved by evolution. Is everybody with me here? Yeah. And then conversely, if I have a bunch of targets, I can find the motif. How? By aligning them the way that I showed you here. Basically, I have a bunch of regions that are all bound by ABF1. I can be like, okay, well, what's the motif? And I can minutely align them and find the motif. And that's great if the region is found from an experiment known as regulator footprinting, where you basically digest the DNA except for the part that's protected by the regulator. And then what you're left with is, 12 nucleotides that were specifically bound. But most of the time, in your binding experiments, you're going to have like 200 bases or 400 bases. So then you're going to have to search within these regions for common patterns, common sequence patterns within those. Yeah. Then, so what I just mentioned, so basically regulator footprinting basically digests the DNA and cuts it out, as opposed to ChIP-seq, which basically randomly makes cuts every like 500 bases or so, and then pulls down the regulator and then just sequences the rest of the fragment. If you now pull down the regulator bound to the DNA and instead you digested the DNA first and then you did the sequencing, it would be very similar. Make sense? Great. Any other questions? All right. So if you have a motif and you want to find what's bound to it, what you could do is generate many variants of that motif. For example, that motif in a bunch of different contexts, and then try to go fish out the protein that binds it. And then you'll get some proteins, and then you can do mass spectrometry, basically figure out what proteins came out. The converse is much easier. So basically, if you have a regulator to find its motif, you can basically look at, chromatin immunoprecipitation, etc. But usually you don't go directly from the regulator to the motif. What you do instead is that from the regulator, you look for its targets. And that's where ChIP-ChIP and ChIP-seq come in. So you can basically use the regulator to do a binding experiment where you basically use antibody to pull down all of the regions that were bound by that regulator. That gives you the target. And then you need to actually discover the motif. Everybody with me here? Who feels that they're learning something? Give me 5, 4, 3, 2, 1. That's awesome. Great. Beautiful. All right, let's dive right in. So you've now done your ChIP-seq experiment, and you found a bunch of regions that are bound by the same regulator. And you'd like to find the motif. Or you've done an expression analysis of a bunch of regions of the genome, and you're finding that these genes are always expressed together. They're going on and off at the same time in a bunch of conditions. So you're like, well, they're probably controlled by the same upstream regulators. Let's look in the promoter regions for common motifs. Is everybody with me here? So very often, even in these, protein binding microarray experiments, what you're going to get out is a bunch of spots in your microarray that are bound by the same regulator, but you don't get the exact nucleotides. So nearly all of the time, you're going to have to do some kind of motif discovery. Okay? Good. So what can you do? You could basically say, okay, well, let me look for local alignment matches between this region and that region. And maybe my local alignment, because these regions are otherwise completely unrelated, that local alignment might actually catch some common patterns between them. Okay? You could also use some kind of expert knowledge to say what should the motif actually look like. You could find the median string by enumeration. You could basically say, let me try all 5 mers, all 6 mers, all 7 mers, and then look for the 7-mer pattern that is the closest to all of the sequences. And you could also say, well, I'm going to use evolutionary conservation first, and I'm only going to do that search within the conserved region. And of course, none of this is perfect because some of these motifs might be slightly different because of degeneracies in the regulatory code. This region can be bound by either an A or a G. So that basically means that if I look at evolutionary conservation, this might not be conserved. And same for this one, C or T. Any C to T change will basically lead to this region not being perfectly conserved. So you have to be flexible with any one of those. Yeah.
You know that you ever look at the structure of the proteins and try to guess as to what nucleotides?
Yes, there are methods that do that. That's not very common, but there are methods that do that. Okay? All right. So what we're going to do now is basically say, if I know the motif, then I can find the instances. How? By scanning that motif and matching it at every position. Everybody with me here? If I know the exact starting positions, then I can make a better version of the motif. Because if now, instead of starting here, I know that it's starting there, I can basically realign on top of each other all of these positions and just recount the frequency of every position for every base. Everybody with me here? So basically, what we're going to do is use an iterative approach, the same thing that we've done before, to basically iteratively use the motif definition to find the instances, get better instances. And now that I have better instances, use those better instances to find a better motif definition. And now that I have a better motif definition, look for better instances. Everybody with me here? So it's the same exact thing that we've done before. And we're going to use expectation maximization. We're going to basically say, we have a bunch of aligned positions. We can use that to infer the matrix representation of the motif. And if I have a matrix representation, I can basically search for that and then find the best matching positions. Okay? So I'm not going to dive too much into the details, except to say that you can calculate for every motif the probability that a position is a match for that motif. How are you going to do that? You're basically going to do a likelihood ratio. What is the probability that this particular sequence was generated by the motif? And what is the probability that this DNA sequence was instead generated by the background? Okay? So the likelihood ratio is what is the probability with which I could have generated this with motif or the background. Everybody with me here? Yeah. So you can use that to then get a probability distribution at every position. And then comes the interesting part. What should I choose? Should I choose the maximum? Should I choose all of them? Should I choose the maximum, even if that maximum is just slightly above the next one? So then the question is, what do I do when I have the motif and I search it and I find some instances with different probabilities? And the answer is that all of the answers are basically acceptable. You could say, well, I could be greedy and I could be choosing always the maximum. And here, always the maximum is a great answer because all of the probability mass is concentrated in one location. I could instead use the expectation maximization approach, which basically says, I'm going to take all of the matches and weigh them according to their probability of matching. And when I'm updating my motif, I will average all of these positions from that sequence rather than taking the exact letters of that match. Everybody with me here? And you could also use a sampling approach that basically says, I will note that this has, I don't know, 70% probability and this has 30% probability. And I will flip a biased coin that will choose this one only with at 70% of the time, but it only chooses that one, or this one only with 30% of the time. Everybody with me here? And then after you've chosen this 30% of the time, you're going to go as if that was the only and best match. Does that answer your question? Great. All right, 5, 4, 3, 2, 1. Who's with me here? Nice, great. So deep sampling will pick one at random. Here, greedy will always pick the maximum. And expected maximization will basically use both. Here, all the methods will agree. Here, greedy will choose this one, but it will ignore most of the probability mass. Here, keep sampling will choose one of them, maybe slightly lower than the maximum, and then it will rapidly converge to some choice. And then expectation maximization will basically average over the entire set of matches. But then the problem there is that it might have very slow convergence because it's now getting pulled into a bunch of directions instead of just rapidly going down one path. Is everybody with me here? Basically, maximization will say, well, the T in that position, here, there's an A in that position. I'm not quite sure. Let me average them and then search again. Whereas Gibbs sampling will say, well, look, this one only has, some portion of the probability, but now I'm going to just assume that it's a T at that position. I'm going to move forward with that. Okay. Good. So then the question is, how are you now moving up this landscape of possible sequence patterns? So basically, you can think of this giant landscape of all possible motifs, and then you're navigating this landscape by sort of saying, oh, well, maybe there's a T at that position. Oh, maybe there's a C at that other position. Oh, maybe this position is either an A or a T, and so on and so forth. And you're trying to find the best motif, and expectation maximization will always arrive at a local maximum where it started. Gibbs sampling might be more likely to kind of jump, from one location to another, because it will take all of its weight, it will put all of its weight behind one of these instances. And then what you always want to do is have multiple initializations. Sometimes you'll start here, sometimes here, sometimes here, and that's sort of more likely to eventually arrive at the global maximum. Everybody with me here? Who feels that they've learned something? Yes? Awesome. Good. All right. So I'm going to skip some of the details on that. So all of this has been the traditional way to find regulatory motifs. You're basically searching, you're updating, and then again, it's this beautiful machine learning, iterative approach, expecting maximization, the same thing that we saw for k-means, for hidden Markov models and Baum-Welch learning, for motifs in this particular instance, and for many, many other problems. Okay. But more recently, deep learning has provided additional tools for understanding genomes. And the same way that you can build from the ground up representations of images based on pixels being abstracted to lines and edges, eventually to eyes and wheels and noses, and eventually to people and cars and sailboats, then you can do the same thing of representation learning from the ground up, from the pixels to the prediction with some internal representations. And one very popular approach has basically been to use convolutional neural networks to be able to learn motifs from the ground up. How? By basically saying that I will search for a common pattern that I will learn from scratch. And that common pattern here, remember in the deep learning introduction, I basically said how you can build these representations from the bottom up, and you can search for filters or patterns that you can keep matching. And in this particular case, those patterns are sequence patterns that you can basically discover as filters in your deep learning framework. So you can basically say, I will try to, at every sequence of, 50 nucleotides, predict the answer of whether that sequence is bound. Where's the answer coming from? The answer comes from a labeled experiment, where you basically went looking for binding of the ABF1 regulator. You now have all of the sequence pattern, all of the sequence segments that are bound by ABF1 and all of the ones that are not. And you're looking for a representation that allows you to predict whether a region is bound or not. Everybody with me here? And you can do that in many different ways. You can do that with, for example, multitask learning. Instead of just saying, oh, I'm going to predict the binding of one regulator, which is very much similar to the traditional way of discovering these motifs, you could now say, I'm going to predict the binding of 500 regulators together. And I'm going to have a vector of 500 tasks of whether each of the 500 regulators is bound or not. And I'm going to use a common set of representations. And those representations will basically be the patterns of the motifs that I will be using as my dictionary, as my alphabet. Does that make sense? Good. So then I can use that to learn not just the first level of the neural network, but I can learn multiple levels. And that basically means maybe at the base, I will learn entire motifs. And right above, I will combine them already. Or maybe at the base, I will learn motif parts. Like there's a common pattern of binding from one arm of a zinc finger protein. And that will be reused in all of the places where that particular domain is binding. And maybe in this particular motif, I will learn it at the second level that this arm and that arm go together. And maybe that will allow me to now match motifs with variable spacing because I will have learned the building blocks individually. Okay? Can I get a 5, 4, 3, 2, 1? How well are you following so far? That's awesome. Beautiful. All right. So you can use that to now start predicting how well you can recover the binding. And what's really exciting is that you can be much more fancy. Instead of just bound or not bound, you could try to predict exactly the shape of that representation of, binding. You can basically look at very high resolution signal from DNA binding experiments, from, variations on the traditional binding approach. You can look for accessibility and regulator binding at the same time. You can look for histone modification marks as one of the tasks that you're jointly learning for. And that allows you to now be much more versatile in the set of signals that you're going to learn. Maybe some of them will be nucleosome positioning signals. Others will be, initiator factors. Others will be splicing positions, and so on and so forth. Is everybody with me here? Awesome. Good. So this has been using experiments to discover the regions, the common patterns in the regions that are bound by a regulator. So most of the time, I know the regulator, and I'm going fishing for a motif that explains the binding of an experiment I've already done. Everybody with me here? But what we can also do is go a step further and say, what if I don't know the regulator? What if nobody has ever done an experiment with that particular regulator? Can I still discover motifs completely from scratch? Everybody with me here? So how are we going to do that? We're basically going to now use evolutionary signatures to discover motifs de novo. And what's the key idea? The key idea is that if you look in a given regulatory region, you'll find that there's islands of conservation underlying regulatory motif instances. Everybody with me here? But an instance doth not make a motif. An instance is an instance. How do you discover a motif? Well, you need a lot of instances. So there's another way to do motif discovery. Instead of just saying, oh, I'm going to look at all of the regions bound by a regulator, I could instead say, I'm going to look for all of the conserved islands in the genome. And then I'm going to find a set of words, a set of patterns that explain these islands in the most compact way, for example. Is everybody with me here? So that's de novo motif discovery. Any questions? Sorry about that. Thank you. I hate when I lose it. I'm like, So I apologize for the overly emphatic sneezing here. All right. So notice here, motifs are not individually found. They're found a bunch of times in the genome. And the question now is, okay, there's CGG spaced by 11 letters from CCG. Some of the time, the island is going to be part of the two motifs together. So what am I going to do? I'm going to take islands and cluster them and then look for matches? Well, maybe a different approach might be to instead start from the motifs themselves and just search for them in the genome. So what we're going to do is basically say, if motifs are really motifs rather than individual islands, individual instances, then they should be multiply conserved in a bunch of regions together. Okay? So then we're going to look for some properties of those motifs. And starting with the GAL4 motif that's bound by the galactose regulator to turn on GAL1 and GAL10 in this divergent region of transcription when there's galactose but not glucose in the environment, because when there's glucose, MIG1 will shut it off. Starting with this GAL4 motif, we can basically say, where is it in the genome? And we can find all of its instances. These are the little red circles. And then we can say, okay, great. How many of these instances are conserved? Maybe a subset. And now it starts getting fun. We can say, let's compare the patterns of conservation of a given motif that we know about, for example, Gal4, that was previously known, compared to a random control motif. And that's a general principle that you're going to have to use everywhere. In other words, to find features that distinguish something, anything, in a genome, a proteome, a chemicome or something, anywhere in a graph, then you're going to look for patterns that are distinguishing the real, whatever that is, like motif in this particular case, from a shuffled version of that. And a shuffled version of that sounds easy, great. You just need to shuffle. But the question is, how do you shuffle? Because if you shuffle in a given way, you're going to find the patterns that distinguish the motif from the shuffles that you created. If you shuffle in a different way, you're going to find different set of features that distinguish your real signal from whatever shuffle you made using that other approach. Is everybody with me here? So the shuffling is extremely important. It's not just like, oh, I shuffled. Like, no, be super precise about how did you shuffle? Did you maintain the exact position? Did you maintain the information content at every position? Did you maintain the total number of instances that you have in the genome? So for example, if I take CGG spaced by 11 from CCG and I shuffle it to AAA, and I ask how often does that happen? If the frequency with which that shuffled version is dramatically different, either too high or too low, then the conservation patterns are going to be very different. So what we're going to look for is shufflings that maintain some properties of the initial motif. For example, if you shuffle codons, you might want to preserve the dinucleotide frequency so that your signal is really, how's the trinucleotide frequency differing from the dinucleotide frequency? And therefore, you're matching that signal as much as possible to truly get at the true signal. Is everybody with me here? Again, this applies to many, many different places. But in this particular case, we basically said, okay, let's ask how often is our motif conserved in intergenic regions compared to controls. And what we found is that Gal4 was conserved more frequently in intergenic regions. We also asked how frequently is it conserved in intergenic versus coding regions. And what we found is that the Gal4 motif was actually less conserved in protein coding regions than control motifs. So basically, control motifs were slightly more conserved. Gal4 was less conserved. You could be puzzled by that, but maybe it makes sense. Maybe you actually don't want to have regulators binding in the middle of genes. Maybe, that's the reason why this is that way. But basically, the ratio of intergenic conservation to coding conservation was actually quite indicative of real motifs. And then the other question is, if you have divergent genes versus convergent genes, in yeast, regulation typically happens upstream of the gene and not downstream of the gene. So then we basically said, well, if I ask how conserved is it upstream versus downstream, we found that Gal4 was much more conserved than random controls that were the same conserved. Everybody with me here? So what are we doing? We're basically discovering evolutionary signatures that allow us to discover real motifs and distinguish them from random patterns. Okay? And using that, we can now enumerate 3 GAP3 patterns in the genome. And what we're finding is that indeed, the ratio between the total count and the conserved count is more or less the same for most patterns, except for a handful of patterns, like a much smaller number, a minority, I should say, of patterns that are much more conserved than what you would expect. And what we found is that the Gal4 motif, CGG11CCG, is in fact much more evolutionarily conserved than what you would expect based on the total count of its instances. Everybody with me here? What's really cool is that there's a ton of other things that are also significantly more conserved than you would expect, and these are the blue points. What about this intergenic versus coding? This is coding conservation, this is intergenic conservation. Most motifs, most patterns, I should say, in gray show more coding conservation, but a real motif, like, Gal4, is actually more conserved in intergenic than in coding. Okay? Same thing here. You can basically say upstream versus downstream. And again, you see that GAL4 is much more conserved upstream versus downstream. But there's actually a bunch of other patterns that might be acting downstream of transcription to maybe end transcription or to do targeting of the RNA to some, post-transcriptional localization of that RNA. Everybody with me here? Yes. Blue means significant, according to that test. And, the real part we're going to worry about soon. So now we're basically finding a bunch of blue points, a bunch of significant points that are conserved above and beyond what you would have expected by chance, given this random model. How do we make motifs out of those? So basically what we did here is that we enumerated A, B, C, D, E, F, where any of these positions are A, C, G, or T. And then some gap. But what we need to do now is basically start collapsing together points that are coming from the same underlying motif. So basically, we're going to enumerate a bunch of seeds. We're going to score them. We're going to now have a much smaller set, and then we're going to try to collapse them based on the ones that are coming from the same point, if you wish, in this motif landscape. Is everybody with me here? And that basically allows you to now discover de novo a list of patterns that are preferentially conserved. And what's very exciting is that we could do that in yeast, then we could do that in human, then we could do that in fly, then we could do that in many more mammals, and so on and so forth. So then in every case, we were able to discover an alphabet of regulatory motifs that were effectively recapitulating nearly all of the motifs that had been discovered up until that point, and many, many more that we were then able to validate experimentally. Everybody with me here? Good. Give me a 5, 4, 3, 2, 1. Who's following so far? Nice. You do. All right. So that's de novo motif discovery. This is the global motif discovery using comparative genomics. Now we want to actually build some regulatory networks. So we want to find individual instances of these motifs. Okay. So how are we going to do that? We're basically going to use evolution again, but now using much more stringent tests to ask what is the overall branch length, the phylogenetic branch length over which instances of that motif are conserved. Okay. We want to tolerate mutations. That's simply when, that position can be either an A or a G, and that's fine. We want to tolerate movement when there's insertions in the alignment, but these gaps are meaningless when you look at any one species because the tree shrew in this particular case has still a contiguous motif occurrence. And we also want to not worry so much about short branches where the additional information provided by these short branches is relatively smaller. But if I see a long branch, that basically tells me that that's much more likely to be real, whereas conservation in a short branch is not as informative because there just hasn't been enough mutation. So we don't care about, short branches. So one way to do that is to basically search every sequence independently and then ask over what total phylogenetic breadth or length or depth has this motif been conserved. Okay. And that gives us some distribution of branch length scores. So basically, if I search a motif, I can basically say, well, this was conserved over a branch length of 0.2 random substitution per site, of 0.9 substitution per site, of 2 substitutions per site. How can you have 2 substitutions per site? This is weird, right? Like basically 1 substitution, I should be able to tell. And the reason is that you have many, many species. And therefore you can kind of see on that tree that there were 1 here and 1 there. Does that make sense? So basically you can kind of add up more and more and more species where the total branch length is capturing the, the random expectation of having multiple mutations on the same tree. Everybody with me here? So we have that now for a given motif. But the most important part is that we need to create shuffles that are very motif specific to be able to evaluate how much more conserved is that motif than I would expect by chance. Why? Because if a motif of length 20 will be mutated much more often, and a motif of length 3 is much more likely to be conserved just by chance. So then the question is, if my real motif is length 3, then my shuffles better be length 3 as well. If my real motif is length 20, then my shuffles better be length 20 as well. And much more specifically, what I want is to have the total number of instances in the genome for that particular pattern. So basically, we have to be very cautious in how we generate these shuffles. And for example, we can take a motif and produce 100 shuffles of the original motif. And we're going to filter motifs by requiring that they match the genome within, say, 20% of the original motif. Because if I change all of the G's with A's, for example, then it might match much, much less in the genome. You don't care about conservation. You don't want to be filtering for your own signal. You don't want to sort of filter motifs that are conserved in a different fashion. No, that's the signal you're trying to catch. But what you're trying to correct for is just simply nucleotide frequency and nucleotide composition. Is everybody with me here? So basically, you're going to produce hundreds of shuffles. Then you're going to filter, requiring that they match the genome, roughly the same amount. You're going to sort potential control motifs based on their similarity to known motifs. You don't want your shuffles to be matching real motifs, so you're going to exclude those. And then you're going to cluster potential control motifs and take at most one representative from each cluster. You don't want to be randomly rediscovering 30 different versions of the same exact sequence and therefore be highly biased. So you want to basically cluster them and exclude the ones that are very similar to other ones so that you have very different control motifs. Everybody can hear? And what's really cool now is that you can basically take your real signal, you can take your shuffled motifs, and at every branch length score, you can basically ask how much more conserved is it than what I would have expected by chance. And that ratio gives you the confidence with which a particular instance is real. The further out you go to the right, the more dark blue there is compared to light blue, and therefore your confidence increases. And then when you're out here, your confidence is just very, very low. Okay? Because just by chance, a branch length of 0.5 doesn't really tell us much. But a branch length of 2.5 is like, wow, that thing is real. Who's with me so far? Give me a 5, 4, 3, 2, 1. That's awesome. You guys are amazing. Okay. So now we're going to basically compute the enrichment of background versus foreground. And what we see is that as we increase confidence for transcription factor motifs, the higher confidence, the more likely they are to be in promoters. And for microRNA motifs, these are acting post-transcriptionally. These are short RNAs that bind the transcript itself. They're much more likely to be in the 5' untranslated region, basically right after the stop codon of a protein, but before the stop of transcription. Okay, so on the mRNA molecule. And that's kind of reassuring, basically says that this confidence is, catching something real. And why is this important? Because therefore we can have a single language to talk about motifs of very, very different sizes. And that's why I can use confidence to basically see it doesn't matter what your size is. A very long motif and a very short motif will basically simply mean the same thing when I talk about their confidence level. These underlying curves are going to be different. For a short motif, they're going to be much further out. For a long motif, they're going to be much further in. But the confidence is going to be a very similar looking curve. Everybody with me here? And then whatever, like that confidence will be higher here and higher there for longer versus shorter motifs, but that projection onto the confidence axis will basically be something that I can use universally for all motifs. Everybody with me here? Awesome. So we can basically now use that to start discovering individual targets of those motifs. Okay. So I'm going to skip this section. We're going to cover it at a later lecture. And what we now have is the ability to start predicting individual instances of motifs. So what have we learned about? We basically learned about regulatory motifs. We learned about this iterative refinement to discover motifs. We learned about Gibbs sampling as a way to go out and sort of follow one of these paths in the distribution by putting all of my weight in a randomly chosen location. We learn about evolutionary signatures for de novo discovery of motifs by taking effectively these islands and clustering them, or conversely, by finding evolutionary signatures of motifs like intergenic versus coding conservation, etc., and then clustering those motifs. We looked at how we can use these evolutionary signatures to discover individual instances of those motifs. Okay, who feels that they've learned stuff? 5, 4, 3, 2, 1. Nice, great. All right, so now let's look at how we can analyze the resulting networks that we get from knowing that a regulator binds here and this other regulator binds there, etc. Basically, what we now have is a series of networks or a series of graphs of connectivities. And we would like to start analyzing these networks. So we're going to switch to how do we do network analysis? And we're going to do that not just for regulatory networks, but for networks more broad. Everybody with me here? So first, we're going to look at all of the different types of network types: regulatory networks, metabolic networks, signaling networks, interaction networks, functional networks. We're going to look at both the Bayesian, probabilistic, and the algebraic views of these networks. Then we're going to sort of discuss a little bit about network centrality, do some linear algebra review to understand about eigenvalues, singular value decomposition, long rank approximation. Then we're going to look at principal component analysis. And then we're going to look at some modern machine learning in networks, introducing graph neural networks that you guys are going to be seeing again in the subsequent lectures. And we're going to probably skip that part. Okay. So their organization of living systems is in layers. At the base, we have the genome. And the genome basically is modulated by the epigenome, which we learned about in the last 2 lectures. And the genome also generates the transcriptome, basically by expression of mRNA, microRNAs, etc., that we talked about in the 2nd week of class. And then the transcriptome itself generates the proteome, which is the translation. Okay, so you basically have transcription, translation, and then modification. Everybody with me here? And we can now start thinking about regulatory networks at every one of these levels. We can start thinking about gene regulatory networks, which is what we've been focusing now, with pre-transcriptional regulation of which regulator is going to bind what region and activate what gene. But you can also think about post-transcriptional gene regulation with the microRNAs that we alluded to briefly, that control the expression of genes, or at least the stability of genes, the degradation, the localization, etc., after they've been produced. You can also have protein-protein interaction networks of basically proteins that sort of bind with each other and then together help each other bind, as I was mentioning earlier, or signaling networks, where a protein will basically bind another protein phosphorylated, and that phosphorylated form now will have other properties, and it will sort of phosphorylate the next one, and so on and so forth, eventually leading to transcription and this signaling cascade. Is everybody with me here? You can also have metabolic networks where you're asking how are different metabolites transformed, from one to the other, and what are the enzymes, and these are the edges that help transform one metabolite to another metabolite. Everybody with me here? So there's very different types of properties for each of these layers. A regulatory network is usually a directed and signed network. If regulator D controls gene A, that means nothing about whether A controls D, usually not. Moreover, there's activating edges and then there's repressive edges. Everybody with me here? So this is both directed and signed and usually weighted. B might turn on C much more strongly than A might turn on C. Okay? A metabolic network is usually bidirectional, where there's an enzyme for turning A into B, and very often there's another enzyme for turning B into A, but not all the time. And so on and so forth. So basically the edges here are the genes and the nodes are the metabolites. Whereas before, the edges were regulatory action, and the nodes were the genes themselves or the regulators. Everybody with me here? Awesome. For a signaling network, we basically have a directed and unweighted network, because after I've phosphorylated something, it doesn't matter how much I've phosphorylated it, it is just phosphorylated. Okay? So it's a directed and unweighted network. For protein-protein interaction networks, these are unweighted and they're symmetrical. If A interacts with B, B interacts with A. Everybody with me here? And then we can also have functional networks rather than physical networks. For every one of these networks, the edges mean something. Here, the regulation means that there's a physical binding of that regulator onto that piece of DNA. Here, there's a physical modulation and sort of change of A into B. Here, there's a physical binding of every protein during that signal. Here, there's physical interactions. But here, a co-expression network simply says that C is co-expressed with D. It's just a functional relationship. It doesn't need to be that like C and D ever meet. It could just be that there's different regulators that control them both at the same time. Everybody with me here? Awesome. And then these networks are undirected, but they're weighted. The strength of the correlation between D and G could be very different than the strength of the correlation between E and G. Everybody with me here? 5, 4, 3, 2, 1, who's following? Nice. All right. So, and of course, there's a ton of information exchange between these networks. For example, as I mentioned earlier, the signaling cascade could eventually lead to the regulator turning on something. And that something might itself be a protein that then forms a complex with other proteins and then come back and actually controls some other gene. And, you could also be transcribing through the regulatory network an enzyme that will then catalyze the rate of change of C into B. And that metabolite itself could be the signal that gets received by the signaling cascade to turn off the regulator that makes more of that metabolite, and so on and so forth. Is everybody with me here? Great. So then there's many different challenges associated with networks. Basically, as I mentioned earlier, one challenge is how do we find the regulators? How do we find the motifs? How do we find the target genes? And we talked about that until now. But then we can also ask, how are the networks themselves predicting cellular activity? And that's where the field of graph neural networks comes in, where you basically want to learn representations on your network to predict some function of the entire network. So for example, for your chemical, you might know that this molecule has some detrimental effect on E. coli that helps the patient. So if that molecule kills E. coli, that's a property of that molecule. And you want to now be able to predict from the graph of that molecule that it will have an effect on E. coli. Everybody with me here? So you want to be able to predict a property of the network. And that property might be, that might be associated with a chemical. It might be associated with a protein, that this protein has a certain function. And that function could be given by a gene ontology hierarchy. That protein is a regulator. That protein is a signaling molecule or a signaling protein. That protein is metabolite. Is a metabolizer, and so on and so forth. Is everybody with me here? So basically, if I have the ability to predict the function of that protein from the graph of its structure, then I'm learning some kind of representation on the network itself. Okay, and that's where graph neural networks come in. I might also want to infer the networks from the functional data. So for example, I might have expression patterns of hundreds of genes, and I want to infer either the structure of the network of, oh, well, A is correlated with, X and Y, and B is correlated with X and Y with some other level. Or I might want the actual functional implication of what is the function that is being computed. So Y is 3 times X plus 2 times A. Okay. And I might also want to understand the structure of the network. For example, what is the most vulnerable place where a virus might be attacking the host, or a virus might be attacking the bacterium? and killing it and so on and so forth. So basically this structural analysis can tell you about hubs that are sort of points of very high connectivity, or about patterns of connectivity known as network motifs. So we talked about linear motifs, which are sort of, A, G, G, A, A, C. For example, that's a pattern that I can kind of look for everywhere. And a network motif might be two regulators controlling the same gene. That's a three-way motif and perhaps interacting with each other. Okay, that's a different motif. Or, A regulates B, and B regulates C, and A also regulates C. That's a feed-forward loop. Or A regulates B, and B regulates C, and C regulates A. That's a feedback loop. Everybody with me here? So basically, these are network motifs. And I might also want to discover functional modules. I might want to sort of decompose a very highly connected graph into subsets that are more densely connected than the rest. Sounds good? All right, so then we talked about physical networks where the edges represent some kind of physical interaction, and relevance networks or functional networks are simply where the edges simply represent how similar things are to each other, how co-expressed they are, as I mentioned earlier, where you don't need a physical instantiation. You can just have some kind of functional interpretation. You can also have probabilistic networks that basically tell you about the independence properties of nodes relative to each other. So for example, if I have 1, 2, 3, 4, 5, 6 variables here and 5 variables there and 3 variables in the middle through which information flows, I might have some dependency of S1 and S3. In other words, if I observe S3, then that changes my posterior probability of S2, which in turn changes the posterior probability of a node in S1. Everybody with me here? So there's information flow. By observing something here, I get some information flow there. But now I can say, if I have fully observed each of these variables in S2, like, V, W, and S, fully observe them, then changing this value here has no more impact because I fully observed those. And therefore, S1 and S3 become conditionally independent having observed S2. Okay, so I can reason in these probabilistic networks about relationships, about information flow, about conditional independence, and so on and so forth. Raise your hands if you're with me. 5, 4, 3, 2, 1. Nice. Lovely. All right. So now we have these networks. Let's have some fun with them. So basically, we're going to represent them as matrices. So I can represent this graph here, where A is connected with B with a weight of 1.5 as, A is connected with B with a weight of 1.5. And this is an undirected network, so that adjacency matrix is going to be symmetric. Everybody with me here? And then same thing for the weights there, and so on and so forth. Now I can actually calculate the degree. What is the degree? The degree is the number of edges that are connected to each node. So for example, node B has a degree of 3. Node E has a degree of 3. Node D has a degree of 2. Everybody with me here? And in a weighted network, the degree could just simply be the sum of the weights of all of the incident edges. Okay? So basically, the degree of B is simply the sum of all of the edges that touch B. And that's in an undirected network. In a directed network, then I want to distinguish the in degree versus the out degree. And then I can talk about in hubs that basically aggregate all of information, or out hubs that propagate a lot of information. And something could be an in hub, but not an out hub. Or something could be an out hub, but not an in hub. Everybody with me here? Awesome. So then using this matrix interpretation, we can now have a lot of fun. We can basically say, if I apply the matrix A on a vector of starting weights, then that basically tells me how those weights are going to propagate through the network by multiplying them through this matrix. So basically, every matrix is a transformation on a vector. So this matrix here is a transformation on that vector. And that matrix basically tells me how will signal flow on this network if I have initial weights of, I don't know, 3 times this metabolite and 2 times that metabolite and 7 times that metabolite. I can basically say, given the metabolic reaction rates given by these weights here, if I have 7 here and a rate of 2, then that will propagate some amount of that initial condition to the next level. Can I get a 5, 4, 3, 2, 1? So how well you're following here? Good. So every matrix operation, every matrix multiplication on a vector tells me how that network, how that graph will change that initial vector. Okay? So the vector could be simply all of the initial weights at every position, and the matrix operation tells me how those weights will be changed by applying that transformation. And it's kind of cool because I can apply that transformation once, that's A times the vector, or twice, that's A times the vector. Everybody with me here? So basically, I'm applying A once, and I'm applying A again, I'm applying A again. But I can also think about the transitive closure of A applied to itself a bunch of times. And that's basically a matrix multiplication that basically tells me that's the effect of applying 3 iterations of that transformation. Okay, so then I can collapse A into an A cube operation, which I can then apply to any vector. 5, 4, 3, 2, 1, how well you're following now? Nice, awesome. Okay, so what eigenvectors are about is a matrix operation, a transformation of that vector that results in simply a scalar multiplication of that same vector. So every matrix shifts the space onto which it's applied, but that shift sometimes will result in some points just becoming twice as long, or some vectors becoming twice as long, or be multiplied by minus something. Okay, and these are the eigenvectors of that matrix transformation. So every matrix is a transformation of a multidimensional space, and then an eigenvector is simply a transformation of that vector, which becomes simply a scalar of that same vector. Okay. Now, why do we care about eigenvalues and eigenvectors? Because they allow us to decompose a set of genes that might be highly correlated with each other into a set of eigengenes. And now, instead of basically saying, I'm going to worry about how every single one of these 40 genes in the same pathway are affected by this experiment, or by this condition, or by the change between brain and liver, then I can say, I'm wondering how that entire module of genes is affected. Okay, so I can reduce the total representation of my regulatory network from hundreds of parameters to dozens of parameters, because now I'm looking for these common patterns of change. Is everybody with me here? Yes? Can I get a 5, 4, 3, 2, 1 on eigenvectors? Great, awesome. So basically, you can basically think about this geometric interpretation of these transformations. So I can basically think about a matrix as transforming the entire space. But instead, if I start decomposing that matrix, I can basically start thinking about, oh, well, that matrix is nothing more than a set of scalar operations to a transformed space. So what this is basically doing is saying, okay, let me rotate the initial matrix, and then scale the initial matrix, and then rotate that initial matrix. And that allows me to now start thinking about any kind of operation. So I can take a matrix, and instead of just saying, here's how that matrix changes the entire space, I can say, well, that matrix is simply rotating and rotating some transformed space where that transformation is just some scaling. Okay, so that's what eigendecomposition is. Instead of just saying, okay, this is a complex transformation on a set of vectors, I'm going to say, well, if I map it to some other dimensionality, and then I map it back to from that dimensionality, in that dimensionality, this operation is just a scaling. Can I get a 5, 4, 3, 2, 1 as to how you're following here? Beautiful, awesome, good. So now we can use that to start discovering the principal components of variation. So if I have a bunch of gene expression patterns, I can basically say, well, they vary along variable A and variable B. But if I transform that space and I rotate it, then I get a much more meaningful space of variation. Instead of just saying, oh, it's the expression of this gene, I can just say, well, it's the combination of expression patterns of these 20 genes here minus those 20 genes there that actually corresponds to the amount of heat shock stress on the cell and the amount of nutritional stress from metabolic starvation, etc. And that heat shock stress versus metabolic stress could be the two dimensions of real variation. And all of these genes are correlated with those underlying true dimensions. Is everybody with me here? And that's what this eigenvalue decomposition allows us to do. We can basically find these eigenvectors in some subset of matrices. And more broadly, we can find singular values for a much broader set of matrices that don't need to be square. They can be rectangular. Okay? And sort of the way that you can think about those is that you can basically do the necessary transformations to be able to apply singular value instead of eigenvalue. And you can use now this PCA to start, or this principal component analysis, to start decomposing this very big expression matrix into smaller combinations of eigengenes and eigenexperiments, if you wish. So basically, instead of just thinking about, oh, well, there's the left ventricle of the heart and the right ventricle of the heart and the atrium of the heart, etc., you can basically think of them as some eigen representation of heart with small variation between them. And then instead of just saying, well, there's this lipid path, this lipid gene, that other lipid gene, and that other lipid gene, you kind of start thinking about the lipid eigengene. Is everybody with me here? And you can basically start now reasoning about a much reduced representation of that underlying space. And all of that is a linear projection, is a linear decomposition of that space. And you can basically do that with the entire space, or you can do that by forcing it to be sparse. And you can basically force it to be sparse by using this sparse PCA. And you can also start thinking about nonlinear dimensionality reduction. Instead of just thinking about the original space as finding a subset of linear combinations, you can basically say, okay, what if I use some kind of neural network with a bunch of nonlinearities to transform that original space into a reduced representation space? Can I perhaps capture more of that variability? So one example is I can take characters like handwritten digits, and I can use principal component analysis on these handwritten digits based on these pixels. And that gives me some kind of separation between, what, for example, 0 might be here, and then, I don't know, 1 might be here, and you can see that they're very different from each other, and maybe 8 and 9 are kind of on top of each other here because they kind of look the same. But you can see that there's a lot of overlap in this space. Instead, what you could basically say is, I want these digits to be as separated as possible. How? By forcing the neighbors not in a two-dimensional space, but in a 1,000 dimensional space, which is all of the pixels of the image that I have initially. And in that 1,000 dimensional space, I can look at the original pixels, or I can look at the representations that I can build on these pixels. And these representations can basically be nonlinear transformation that can recognize edges and shapes and corners and loops and you name it. Is everybody with me here? So by allowing a nonlinear transformation of that space, I can basically force the additional representation to allow me to separate things out more strongly. And in particular, I can basically start thinking about these very high dimensional relationships, the relationship between pairs of points in this very high dimensional space, and asking, how can I preserve them in a lower dimensional space? Okay, let me explain. This is basically known as t-statistic stochastic neighbor embedding. Neighbor embedding basically means that I'm going to embed the relationships of all pairs of neighbors. Stochastically basically means that I'm going to adjust the parameters to map this high dimensional space into lower dimensional space progressively. So I have this very high dimensional space where point red here is very close to these 3 blue points. And I want to find a transformation from this high dimensional space, a nonlinear transformation that preserves the relationships of my 3 neighbors in the high dimensional space, my closest 3 neighbors in this 1,000 dimensional space, are the same as my three neighbors in the two-dimensional space. Is everybody with me here? And what I'm looking for is some transformation of the original space into a lower dimensional space that preserves the relationships between pairs of closest neighbors. So basically, I have a very high k-nearest neighbor graph, very high dimensional k-nearest neighbor graph, and I'm projecting into a lower dimensional k-nearest neighbor graph such that the pairs of distances are preserved, okay? But that transformation can be very highly nonlinear, okay? And sort of what I care about is this property of maintaining that dimensionality, but in a much sort of condensed representation. Basically, this is what this actually looks like. You can basically start with a set of points, and then, you know, it sort of starts going through a bunch of steps, and then it sort of adjusts this this dimensionality, and then it can reveal patterns of these points. Okay, and that's basically what lies at the core of all of the single cell data analysis that we showed you earlier, where you're basically taking the 20,000 dimensional long vectors and you're condensing them into a two-dimensional representation. Am I going to be doing distance analysis here? No, this is just for visualization. This is just to give you insights. But what I'm going to be doing is doing the mapping of this very high dimensional space into these lower dimensional spaces. And then I can show the expression of a given gene into that lower dimensional space. Okay? And then the last part is that you can now start thinking about learning on these graphs directly. So basically, these networks can be anything. They can be, you know, gene-gene interactions. They can be physical networks. They can be parts of an image. They could be different words in a text that are connected to each other. They could be networks of genes or of people. They could be generative models. They could be molecules in 3 dimensions, where the edges not only mean connections, but they also have angle constraints on them in geometric deep learning. And what I'm trying to do is learn representations of these molecules or of these, you know, physical relationships or of these texts that capture the properties of the corresponding object. Okay, and that's where learning on graphs comes in. That's where I can basically take a graph and carry out predictions using machine learning on these graphs at the node level, where I'm using the nodes and the properties of these nodes to predict some property of the entire graph by learning progressively more abstract representations. Basically, I might have some biological network, I might have some graph machine learning model, and I will learn representations of either individual nodes or representations of individual edges or representations of subgraphs, and ultimately representations of the entire graph. And the way that we're going to be doing this is that in the same way that we went from pixels to edges, to corners, to shapes, to objects in pictures, now we're going to be doing it at the level of subgraphs, where individual nodes are going to have a property, and then I'm going to update the property at level 0 representation is going to be the property of the node itself. At level 1 representation, that property will be influenced by all of the neighbors. At level 2, by all of the representations of the neighbors, which are influenced by the neighbors themselves. So at level 2, it'll be the neighbors of my neighbors, and so on and so forth. And I can do that at the node level, at the edge level, and all of that, you know, I can then benchmark as to, how well I'm doing. I can sort of start thinking about explainability, and so on and so forth. Okay? So that's the lecture for today. So we basically talked first about, we talked first about how to infer these networks using the building blocks of the physical motifs that are bound by the regulators, and how we can discover those through either expectation maximization, enrichment, or evolutionary signatures. And then we looked at how we can take the resulting networks and start analyzing properties of these networks, such as, you know, centrality or eigenvectors or principal components or lower dimensional projections, and how we can actually do machine learning on these networks. And this is the segue to the next 2 modules, where you're going to be learning about all of this in the context of first protein structure and then chemical structure. So all of what I showed you in the very last part is what we're going to now understand much more deeply over the next few lectures, but I wanted to sort of give you a segue into all of that. Who's excited? Awesome, great. So thanks again, and please come to office hours. That's how you're going to do P-sets. So start early. You've had, I think, 2 weeks already. So start early, and then, you know, make progress, and then come and ask questions. Thank you.